All right, welcome. Hopefully this will be the last video here. We will be entering the realm of gene expression and gene regulation. So gene expression is really you're copying your DNA into RNA, which then is made into a protein, right? So you're really trying to go from your genomic information, like data storage, into something useful and physical and productive, like a protein. There's two main steps here, transcription and translation. So transcription is really you're copying the DNA into a complementary sequence of RNA, right? So DNA, deoxyribonucleic acid, right, is a nucleic acid. It's in the name. RNA, ribose nucleic acid, right? It's also nucleic acid. Also, all the cell is really doing here is it's pulling out its pen and paper and writing in English from the textbook onto the piece of paper, right, in English. It's going from the same language to some other mechanism, right? Because say you're in the library, you need to make a copy of a book, but they won't let you check it out. So what you do, you take the book to the printer, put it on the printer, and hit copy, right? And now you have a sheet of the identical copy of that page that you wanted. That's what the cell is doing here in transcription. It takes DNA, copies it pretty much identically into a complementary sequence into that RNA molecule that is now easily movable from inside the cell nucleus to outside the nucleus, right? This molecule is called mRNA. There's different types of mRNA, but we call it there's different types of RNA. One example of that is the mRNA because it's the messenger RNA. And these messenger RNAs are actually what are going to be translated into protein. There's other types of RNA like we saw with ribosomes. There's ribosomal RNA. That does not make a protein, okay? It's the mRNA that makes the protein. And we're talking about gene expression, right? So mRNA is translated into protein. No other type of RNA does that, okay? So when our mRNA moves from the nucleus to outside the nucleus, it goes to the ribosomes where it's translated into a polypeptide sequence, right? Because now you're going from a nucleic acid to a amino acid. So you're translating into a different language, right? So you've made the physical copy of the textbook in the library onto a piece of paper, but now you're in Spanish class because everyone has to take a language class at ORU, and you have to translate the English copy page that you just made into Spanish, right? That's what the cell is doing here because Spanish is the actual, paper that you just translated is the assignment that you have to turn in, right? That's what you're supposed to do. That's the functional part of that homework assignment. The protein is the functional part of gene expression, right? Because proteins are really the molecule that the cell is making to do most of the cellular functions. So gene expression is two steps: transcription from DNA to RNA, and then translation, which takes the mRNA that was produced and turns it into a polypeptide sequence or a protein. So it's more specifically here what that looks like. So transcription, what you're taking and reading is the DNA, which is nucleic acid, and making it into a messenger RNA. There is at the gene that you're trying to make, because in the cell, a protein is produced or read by a gene. That gene encodes for a protein. When that gene is transcribed into mRNA, it has to know where to start that, copy process. Basically, what page am I copying? Page 1 or page 1000? There's a big difference. So that transcription start site tells the mechanisms and the proteins involved in this where to start making the copy. And of course, there's an end or a terminator that tells it when to stop making the copy for that specific protein. It's mRNA. So basically, what page number do you need to copy to? Now, when you're copying DNA into mRNA, remember there's all those nucleotides in DNA, but what's unique with RNA is that instead of a T, you replace it with a U. So instead of thymine, it's a uracil. So adenine will only bind with a uracil in RNA, and then C's only pairs with G's, right? That's as same with DNA. So one way when you're looking at a nucleic acid sequence to determine whether it's DNA or RNA is there a U instead of a T, right? Because that gives you a key. Hey, if you see a U, then you know it's RNA. If you see that T in that sequence, then you know it's DNA. When you make that initial copy, it's called pre-mRNA because it needs to be slightly modified and needs to be processed and undergoes a maturation step here to make into a functional mRNA sequence there. But that's just a thing we're not going to worry too much about, but just so you're aware that there is more steps than just making it into DNA, into the mRNA. Now, when you have the mRNA molecule now, it's complementary to the DNA. When you look at it, you'll just see the A's, U's, C's, and G's. But when you look at them, when you're translating into protein, we'll see it really best in the next slide here. But the mRNA is really what it's containing is codons, which is sequences of 3 nucleotides that specify which amino acid should be added at that step during the synthesis of the new protein molecule. There are 64 different codons, but there's only 20 different amino acids. So there's some redundancy there built into our genomic code, which I think is a beautiful sign of an intelligent creator, because what happens if there's a slight genomic mutation? Well, now you have some redundancy so that, you know, your protein isn't going to be incorrectly made and potentially kill you, right? Or cause cancer or so forth, right? There's redundancy there so you can still function and live and flourish. So this is kind of a very brief description of transcription. In class, we'll probably go a lot more in depth on this just because it is a more complicated function, and I do think you guys need to understand it now that we're in the 21st century where nucleic acids technology is really having a boom cycle right now, especially in medicine. So you guys will need to know this as much as possible. Translation. So remember, we're translating from a nucleic acid language, which is our mRNA, into a polypeptide sequence, which is our amino acid language. This occurs on/in ribosomes, right? And it takes the mRNA, copies it into a polypeptide sequence, right? In translation, there's 3 different types of RNA involved. rRNA, which is the ribosomal RNA, which remember helps form the structure of the ribosome. There's tRNA that carries the complementary anticodon sequence to the 3 nucleic acid nucleotides in RNA, because what that tRNA is carrying is the amino acid to be added and helps identify which one should be added, and then there's, of course, the mRNA. So the tRNA has those anticodons that complement the mRNA, as the image shows here, because it will identify and carry in the correct amino acid to that codon sequence, okay? Because inside, once it gets into the ribosome catalyzes the reaction, forming a peptide bond between the previous amino acid and the new amino acid being made, okay? So this translation is happening all the time. It's a big energy requirement for the cell, so it's a pretty important process, and it helps keep you alive. It makes you you. It makes your eyes blue and or brown or green or purple or yellow. You know, that's all protein based. So you know that's where it comes from, is gene expression. So that was a very quick overview of gene expression. Like I said, we'll probably go cover it again in lecture in person just because it can be complicated. Don't want to get too lost there. Now, cell cycle is another important thing that has to do with the DNA, because cell cycle, right, is just replication, division of the cell, either mitosis, where one cell just makes an identical clone of itself, or meiosis, which is required for sexual reproduction, right? But all of this is engulfed in the cell cycle. A lot of the time, the cell is spent in interphase, which is a phase where the cell's not really doing anything other than replicating DNA and doing normal cellular activities, right? Which, as you can see, that's most of this graph here is interphase. There is G0 phase, which is this even more, I guess, sleeper stage, a quiescent, senescent stage. Now, when we talk about actual cell division here, that's this in this yellow is mitosis. You could just replace this with meiosis. But, you know, it's a very small sliver of the cell cycle. The cell cycle, really interphase is broken up into 3 cycles here, G1, S, and G2. Each step has different things occurring. And then you have your M phase, right, which is mitosis or meiosis, but in this example, it's mitosis, where you have 5 other steps occurring, such as prophase, metaphase, anaphase, and telophase, and cytokinesis, all of which is the idea here is you're splitting one cell into 2, so you're making 2 daughter cells out of a mother cell, which are genetically identical. So interphase here, it's really the preparation and normal stage for the cell. But if it's starting to get ready for cell division, what happens is the cell increases in size and wants to store lots of energy because it takes energy to do this. It needs more organelles because you need mitochondria, Golgi, ER, rough ER, vesicles, you name it, ribosomes. It needs to make basically double it, right? So it has to increase in size, replicate those organelles. It needs to make a whole brand new set copy of DNA, right? And then those centrioles, which are microtubules that pull apart, that are required for the cytoskeleton, right? It has to make duplicates of those. So everything in the cell it has to make duplicates of. So that's why it has to prepare for it. Now, the G1 phase, which is the first phase, it's really carrying out these normal activities, increasing in size, replication of organelles. S phase is where DNA is really predominantly synthesized, and DNA is replicated. Nucleus gets larger. And then the second gap phase is preparation for the division. It's doing a number of checks, maybe making more, so the cell can prepare for its division. There's that G0 phase, and that it goes into rest here. So that's where a lot of these phases are not occurring. So it's just kind of going into a sleeper stage. So that's the interphase step there. Now, cell division here, the steps of mitosis are prophase, prometaphase, metaphase, anaphase, telophase, and really I include cytokinesis in this. Mainly from the fact that if you don't have cytokinesis where the cells cut apart, then the cell still dies from lack of cell division. So prophase is where the chromatin condenses and those centrioles migrates to each pole of the cell, so either end of the cell, where the chromatin's condensing into your standard X-shaped that we envision chromosomes to be. So that's what we mean by condensing. Then prometaphase is where those spindle fibers, right, so another form of microtubules, extend from our centrioles. They bind to the center parts called centromeres of the chromosomes, and bind to a sequence called kinetochore on those chromosomes, and starts organizing the chromosomes linearly in the cell. Because what's going to happen is in metaphase, the spindle fibers, the microtubules attached to the chromosomes, align them vertically in the middle of the nucleus. And then in anaphase, the spindle fibers start pulling apart and splitting the chromosome in two, because chromosomes are identical copies. There's two copies, one from dad, one from mom. It splits them apart and moves them to opposite poles, and then cytokinesis begins. Because then after that, in telophase, once you split apart enough, you form two nuclei, and then the DNA starts to decondense a little bit. Your nuclear envelope reforms, and then you have the rest of the cytokinesis, the cutting of the cell, right, to where you fully cytokine. Yeah, I'm trying to make another verb of it, but we have further complete separation of the two cells. So inside of that, here's a figure here of your DNA replication. So you need to make a copy of the DNA, but it's semi-conserved in the sense that you will copy one old strand, will produce a new strand that's bound to it. The other strand that's complementary to the other old strand makes its own new copy, which is bound to it. So that's how you make two copies. And during DNA replication, you make a new complementary strand of DNA that's bound to one old or mother copy or template DNA strand. So here's the example of what I was talking about in that anaphase of separation of the chromosomes, where you have your chromosome, mitotic chromosome. You have your centromere where the microtubules bind to, and then during anaphase, when it pulls them apart, it pulls these two apart, because these are copies. These are copies of the DNA. They're technically identical, but they're copies of each. Here is an image of what the cell division actually looks like here. So interphase, building things up. Prophase, chromosomes are condensed into what we think structure is. Centrioles go to different poles. Spindle fibers are formed here in the nucleus. It aligns the chromosomes in the metaphase here. Then in the anaphase, it pulls them apart. And then now you have cytokinesis of the nucleus to produce two different nuclei, and then full cytokinesis to complete two different cells, which is where the cell's then completely done. It goes kind of like into a G0 phase to store up energy again before it has to undergo mitosis again. There is a thing that's natural that's called apoptosis. This is natural. It's really useful. It's ideal for it to occur. It's programmed cell death. So you want to maintain the correct amount of cells, not too many and not too little, especially when you're talking about like healing and scar tissue and that sort of thing. You want to have damaged or infected cells, die, but controlled death. So the cells will undergo apoptosis. And a lot of times the T cells are involved in this process to help contain potential harmful chemicals that are released during cellular death. And like macrophages are here, an example of immune cells that will engulf the cell fragments. T cells do that a little bit as well, but macrophages do a lot of this removal. You want it regulated because there are harmful chemicals inside the cell that can damage the tissue surrounding it if it just explodes, like the hydrogen peroxide. Don't want that going around anywhere. Now, there are effects of aging on cells. There's we don't know exactly how this occurs. We thought we did, but we don't. We're getting closer, but there is a cell clock that we know of. There are apoptosis genes that we think may affect and not work properly, and that's why cells age. DNA damage accrues over time, just from replication mistakes or too many suntans. Free radicals, which are resulting from chemicals that can damage the DNA, and other proteins and other molecules, and then energy damage actually in the mitochondrial DNA or the mitochondria themselves. So we'll end the chapter there. I'm glad you guys made it this far, and we'll see you in chapter 4.